Hi, this is Miss Litton, and this is 7th period HBio review for our biotech quiz. Say hi.
Hi.
Okay, first thing right off the bat, I want to remind you, remember to do your additional objectives. Okay, I believe there are 2 for this chapter. One's on the front page of the calendar, and one, like where as you scroll through it, one's on the second page. So make sure you look at both of those, because I'm going to tell you for sure they are on the quiz. All right? Remember that? You forgot about that? Okay. All right, so the first thing is we reviewed the central dogma. And so you just want to remember that DNA to DNA is called replication. When you use that DNA to build RNA, what's that called? Transcription. And then when you use that RNA to build a protein, what's that called? Translation. Now, in the quiz, I will be applying some of the lab that you're doing currently to the information and kind of weaving those together from the chapter. So what's the DNA in our case in our lab? What's the DNA? Plasmids, okay? So our 2 plasmids, what are the names of our 2 plasmids? Piara and pecan. Okay, and currently as they exist, both of those plasmids have the ability to be resistant to antibiotics, right? Okay, but there is a gene that we super much like, and that is the RFP gene, but that RFP gene is located in pecan, and we can't currently access it, right? It can't be transcribed or translated at this point, okay, because there's no way for RNA polymerase to bind to the promoter. Okay, so what we're trying to do is cut that gene out and insert it into Piara so that the RFP gene can be transcribed into mRNA and translated into a protein, and that protein is going to be red so that the bacteria glows red. You all right with that? Okay. We talked about that when you want to make more of something, that would be a clone. When you make your bacteria that has taken your plasmid in, it will then make more of itself, and those will all be clones of that parent bacterium that you have transformed. We talked about the need for vectors, and viruses are really good vectors, but we're not working with a virus in this lab, right? And plasmids are really good vectors, and plasmids are those extra rings of DNA, and here is one in this cell, and we have Piara and pecan, and those are the ones that we are manipulating, right? Okay, and then we talked about the process of biotechnology, and you're doing something similar. It's just not the human insulin gene, it's this RFP gene. And when you want to get a gene from one location to another, you have to cut that gene out. What do we use to cut the gene? Restriction enzymes. And restriction enzymes cut at a specific series of bases. And what is that specific series of bases called? Recognition sequence. Okay? And each restriction enzyme has its own unique recognition sequence, right? And if I want to mix and match DNA, I have to be able to mix and match. I have to have a similar recognition sequence if I want to combine DNA from 2 different sources, which is what we're doing in our lab, because we use one restriction enzyme, BAM, to make a BAM cut in our DNA, and another restriction enzyme, what? HINDI, to make the second cut. So if I do the same thing with another plasmid, then I can mix and match them. Okay, turn to the person next to you and say, of what she said so far, what do you have questions about? Go ahead, ask her. Okay, so in this picture I'm showing you right here, they're taking this human insulin gene, they're cutting it with a restriction enzyme. They're using the same restriction enzyme to cut open this plasmid and insert this DNA, just like we're trying to insert our RFP gene into a plasmid. Then you take this plasmid, insert it into bacteria. Now you will make clones of this bacterium, all with the ability to have the plasmid in it, and all with the ability to generate that protein. In our case, it's that red fluorescent protein that we're trying to generate. So we talked about restriction enzymes and how they cut. And you could have a blunt cut, which you can't do much with because there's no way to connect, right? But if you do the staggered cut and create these sticky ends with a series of bases, that allows you to mix and match different DNA sources. And the cool thing is how universal DNA is, and how it uses the same RNA and how it uses the same amino acids to build the proteins no matter what organism you're in. So as long as you have the right genetic code in there, you can get that protein. All right, then we talked about here, we talked about you can do it outside of bacteria as well. You could put DNA into plant cells, or you could put DNA into farm animals, right? We looked at all the different ways you could put DNA, and in order to express that particular trait. Okay, do you want me to ask you these questions? Yes. Okay. Why don't we go in as our favorite holiday? Wait, I have to remove. There we go. Go in as your favorite holiday. And I'm going to pause this just for a minute. Guys, totally. Okay, so the question was, these enzymes are needed to introduce foreign DNA into a vector, and all of you got it right. So we need to be able to cut with restriction enzymes, and then DNA ligase is going to seal it. All right, so next we said, okay, how can we take this information of how we can manipulate DNA and use it, who's that? And use it to our benefit. So one of the strategies is when you have a very, let's say you have a crime scene or a very small amount, maybe you're looking at a fossil or something like that, and you can have a little bit of DNA and you want to make more of it. What's the process when you make more DNA? What's that reaction called? PCR, which stands for what? Polymerase chain reaction. What can you do polymerase? When I hear polymerase, I think of DNA polymerase, which makes more DNA, right? Because that's what I want. I want more DNA, so I use DNA polymerase. And I use it in a thermocycler. That's the name of the machine. So you use a thermocycler, right, in order to do the PCR reaction. And it's a sequence of heating and cooling cycles. And if you're going to make more DNA, the ingredients would be DNA nucleotides, right? To build more DNA, I need more DNA nucleotides. I also need some enzymes. What enzymes would I need? DNA polymerase, right? So you need the enzyme that's going to do it, and you need the components to build more DNA than a nucleotide. What's a nucleotide made out of? Sugar phosphate and a nitrogenous base. So you would put in, you would put in nucleotides that contained adenine and thymine and guanine and cytosine. Yeah, and you would use, and you see that in your notes, the DNA polymerase that they use is from an archaea. Remember we talked about the 3 domains. And the reason is we know archaeas are extremophiles, so they can take the hot temperature. Now, why do we need a hot temperature? Why do we have this hot, cool, in a thermocycler where we're alternating the cooling cycle and the heating cycle? Yes, the heat will denature it. So instead of being double, it breaks the hydrogen bonds. Then the DNA polymerase, and you need some primers in there as well, and we'll put the nucleotides in the right spot so you can go from 1 copy of DNA to 2 copies of DNA, and 2 to 4, and so on. So in a matter of minutes, you can get thousands and thousands of copies of that DNA that you can use to test and analyze. Then that process of analysis, when we want a DNA fingerprint, you're familiar with. You do gel electrophoresis. And DNA will sort itself according to its size. The smallest pieces will move through the gel the fastest, and the bigger pieces will move through the slower. And each organism has a particular fingerprint. And you look to the areas, because most of our DNA of all of us in the room, almost all of our DNA is exactly the same. It just has to do with cellular functioning and the building of our bodies and repairing of our bodies. But there are pieces, just like we are unique around the room, there's a very small percentage of our DNA that is unique to us. That is the part that you compare when you're trying to see who committed a crime, right? Because we can't compare the stuff that's the same in all of us, right? We all have to have a heart that beats. You know, we all need to make lipase, right? We all need to make different enzymes that we need in our steps of metabolism. But what you're going to look for is those areas that are unique to each individual. And you run those sections of DNA through gel electrophoresis and sort it. Okay, and when you do it, it looks more something like this. And I want to get to another picture here. So in this case, what they're doing is when they look at that section, they know that the DNA will sort itself according to size, and we found a problem with this picture, right? What was the problem with this picture? You remember? Yes, Jojo.
For like the line on the, that one?
Yes.
Would be farther up?
Yeah, should be here instead of here, right? Because if it has two pieces, a 12 and a 16 repeat, what is this one out here? 12, because it's smaller, it will move. Yeah, and then the 16 will be here, and repeat over here, 12 and 16. So this 12 should line up with all the other 12s. Right, so that's good if you can see that and see how that's wrong and how they modeled that process. They just weren't thinking that through. All right. Then the next thing we talked about was biotechnology. So using natural living organisms to produce a product that you want. And we talked about you could do that in bacteria. And this is transgenic bacteria, bacteria that's been altered to break down oil, or it could be altered to withstand frost, or it could be altered to mine for minerals, or you could alter it to make growth hormones, or in the case of Amgen, insulin or clotting factors. So you genetically engineer that bacteria for a particular product. You can also genetically engineer plants, maybe making them frost resistant or drought resistant, or maybe you make a chimera type plant that has two different species within one. This is not like a tree where I used to have a tree in my old backyard where we had grafted, you can do, I forget what you call it, but it's like, you know how peach have pit? You can put several different, here sweetheart, were you ever worried?
Yeah, I was just walking to my car. Well, no, I didn't notice, but then I know. Never mind.
Did you get a text?
Yeah, Lauren texted me.
Okay, good.
Thank you.
All right. So you can graft like an apricot and a peach, anything that has a pit, and a cherry, you can put it all on one tree. But it's not coming, right? That's something you graft on. This is actually one plant, not grafted, right? But if it could produce both products, that would be pretty cool, right? Or higher nutritional value, or any kind of antibiotics. If you could genetically engineer plants, that's where you want to go there. And then with animals, you might want to clone animals to produce other factors as well that you could harvest, like in their urine or in their milk. And that was it. It's really short. It's really short. And I asked 5 questions. They're all real straightforward, like, what does this do? What does this do? I think it's pretty easy quiz, but just make sure you do your additional objectives, right? Did I say highly suggested reading and thinking? That's because that's what AP does, but you do additional objectives. Yes, sir?
So the only things we're supposed to know from our lab are like relating it to translation and stuff like that?
Yeah, just the big ticket, like how I introduced the lab and like what are our plasmids, what are we trying to do, those kind of things, the basics of our lab. Yes. Any other questions or concerns, my friends? Okay, have a piece of toast. I hope you didn't stay up too late.